All right, so the last thing we're going to talk about with regard to nucleic acids is translation, this last step of reading the mRNA. This is going to involve this machine, which is called the ribosome. Again, a ribosome is like an enzyme, but it's a weird enzyme in that it not only contains amino acids, but it also contains RNA. The RNA in a ribosome is called a ribosomal RNA or rRNA. These ribosomes are bound to what's called the endoplasmic reticulum within the cell. Basically, if we're going to kind of like visualize it, here's my cell. Here's my nucleus. This is where the DNA lives. It's going to spit out mRNA. All right, and what we see is there's these structures. They have a lot of surface area. They're kind of like zigzaggy. These structures are dotted with ribosomes. Ribosomes are kind of like embedded in the surface of this structure. And these ribosomes are here to read the mRNA and produce proteins, right? So a ribosome will take a strand of mRNA, and based on the sequence, it knows that this amino acid should be linked to this amino acid, should be linked to this amino acid, et cetera, okay? Those new proteins that are made are kind of, because the ribosome is just embedded in this endoplasmic reticulum, the proteins that are made in the cytoplasm. That is everything else, right? So everything else, the middle part of that cell is what we call the cytoplasm. All right, so first let's talk about the tRNA molecules. So these are an important component of this whole thing. They are what brings the amino acid to the ribosome and recognizes a complementary strand on the mRNA, okay? So first let's look at a tRNA real quick. You'll often see these 2D projections here of the tRNA. In reality, tRNAs have this weird kind of 3D shape, but they do have this kind of L configuration. Important components. One is what's called an acceptor stem. This is where the amino acid is bound. Okay, so one tRNA will have a valine bound, another tRNA will have an aspartic acid bound, another tRNA will have a leucine bound, et cetera, et cetera. There are all these different tRNAs floating around that have all the different amino acids bound to them, all right? And the amino acid is going to correspond to this 3 nucleotide stretch down here, which is called the anticodon, okay? So each anticodon has a, so let's say that the anticodon, right? Because this tRNA strand, it's just a big strand of mRNA, or I'm sorry, it's just a strand of RNA. So there's 3 RNA nucleotides down here we call the anticodon, and they uniquely correspond to an amino acid. So for example, if all the tRNAs that have an anticodon of like, I'm just going to make this up, this might not be true, but let's say like the anticodon is UUA. Well, those would all correspond to leucine tRNAs. Okay, so there's this correspondence there. And it's going to be the identity, the anticodon, that's what helps the ribosome know what amino acid to add. Okay. Yeah, so I guess step 1, and this is what we talked about last lecture, the DNA gets read and made into RNA. That RNA is specifically mRNA, which codes for the protein, is kicked out into the cytosol. Okay, that mRNA, once it leaves the nucleus, is going to attach to the ribosome. All right, so the ribosome is the machine. The mRNA is the information that tells you what the protein should have, what the protein sequence should be. So the first step is the ribosome binds to that mRNA. Okay. Then now it will sort of continuously bind tRNAs. And the tRNA that binds is because it recognizes that mRNA, right? So here we have a long, let's say that we have a long strand of mRNA, there are these three nucleotide segments, which are called codons, right? So here I have my ribosome bound, and the ribosome's huge, but it's just, ribosome has a large subunit and a small subunit. And so what's going to happen is ribosome is going to look and say, okay, this segment here, let me actually, so large subunit, small subunit, the ribosome is going to look and say, okay, this segment here, these three amino acids are, for example, UCA. So then the tRNA that's going to come along and bind inside that ribosome is going to have the complementary sequence to those three codes, right? To those three amino acids in the codon on the mRNA. So for example, the anticodon on the tRNA in this case would be AGU, and that means that it should be a serine that is added next. Okay, so the mRNA contains the information. The tRNA, when it matches, it will bind and will complementary, those complementary base pairs will match. And then the ribosome says, okay, this is the next amino acid that needs to be added. Okay. The genetic code then on that mRNA consists of those codons, those three-letter amino acids, those three-letter nucleotide sequences. Tells you what amino acid should come next. So there's a different codon for all the different amino acids, for all 20 amino acids needed. Okay, that is what this table is here, and we'll look at that and we'll sort of use this table in a second, but like just for example, UCA, that codon means that there would be a serine that should be next. ACC means that there should be a threonine added next, right? So judging by the sequence of the mRNA, we know what amino acid is going to be added next. Okay, so this is also on your constants page. We'll be able to use this. We'll do some practice here in a second. In addition to what amino acid comes next, there are start signals and stop signals. Okay, so the ribosome will actually specifically first bind to a UAG codon. That's how it starts things off, right? It needs to, the first tRNA that's going to come on is going to come on at an AUG codon, always, all right? There's also stop codons as well. So the ribosome is just going to keep on reading, keep on adding codon after codon after codon until it comes to one of these three codons here. These are stop signals. And they essentially have special tRNAs that once they bind, they break apart the whole thing. Okay, they don't add another amino acid. They'll bind to the ribosome, and then they break apart the ribosome and free that mRNA from there. Okay, so if I gave you a mRNA sequence and said, here are the different codons, and so this is what this page is summarizing here. We'll use this table here in a second, but the lecture slides make it easy. The first three letters would correspond to the first amino acid, so that would be, in this case, CCU is proline, so that would be the proline. AGC is a serine, so that means serine would be next in my polypeptide chain that's being built. Then GGA for glycine and CUU for leucine. All right, so that's mRNA fragment here, codes for an amino acid sequence pro, ser, gly, lu. Right, and it's 3 codons per amino acid. So a nucleotide chain that has, what is that, 12 nucleotides corresponds to an amino acid chain of 4 nucleotides. Okay, so again, this is the chart that we're going to be using. And yeah, so we'll get some practice for that. So real quick, again, the tRNA is going to be, the tRNA that gets selected by the ribosome is going to have a complementary base pairing to those codons, right? So the anticodon on the tRNA will be complementary to the codon on the mRNA. And then what happens is the ribosome will bind one tRNA, it'll recognize the other one should come in based on that anticodon, and it transfers the amino acid chain that's being built to that new one, right? So here's my growing polypeptide chain. This incoming tRNA is selected because its anticodon matches the codon on the mRNA. And so what's going to happen is it's going to form, the ribosome will then catalyze the formation of a polypeptide bond between these two, between the amino acid on the one and the growing polypeptide chain, and then it will kick off that free tRNA. That tRNA will then re-enter the pool of free tRNAs and it will get recharged again, meaning that it will have whatever amino acid it should have added on, right? So after this alanine thing gets kicked, this growing polypeptide chain where the alanine gets kicked off, this is an alanine specific tRNA, so it will just go and bind another alanine amino acid to the top, right? To this acceptor stem right here. All right, so yeah, so again, this is actually a good picture here. You have one amino acid that has, I don't know why they cut it off. This is clearly part of a larger image, probably in your textbook. Let's see if I can find it and point you all to it. Yeah, so this is on page 739 is this very nice big image that shows all of this. The lecture slides have it kind of cut up. But the bottom line is we see our mRNA here, that's this strand. We have the ribosome that's bound to that mRNA, and we have these tRNAs that are coming in that have their anticodons matching the codons on the mRNA. That's how we know which tRNA should come next. Each tRNA, depending on its anticodon, has a specific amino acid it's bringing in. And so that's how the ribosome knows what amino acid to add next. And so eventually this growing polypeptide chain will be transferred over to this tRNA, and the free tRNA will be kicked off the ribosome. We say that protein synthesis happens in 3 steps. So the first step is the initiation step. This is where the ribosome is binding to the mRNA. Actually, first the small subunit binds to the mRNA, and then the large subunit comes on top and kind of traps that mRNA in between. The elongation step is the synthesizing of those polypeptide chains. Different tRNAs, depending on their anticodons matching the mRNA's codons, bind inside the ribosome. The ribosome is catalyzing the formation of that polypeptide bond between the adjacent tRNA molecules. Once a tRNA loses its amino acid, it's kicked out of the ribosome. Then, of course, there's the last step, which is termination, which is what happens when you reach the stop codon. This leads to the binding of a special tRNA molecule, which comes in, recognizes that stop codon, and breaks the ribosome apart, freeing both the mRNA and the polypeptide chain. Okay, so so this is one of the things we want to be able to do here. So given what's in the nucleus, the DNA strand, we have a template strand, which is the one that's being read to figure out the mRNA. And then the other one, which is called the informational strand, I guess, it's just the one that's not being read. Okay, so only one side of the tRNA is going to be read. It could be that they both code for stuff, but at any given time, only one strand is going to be read. So from our template strand, we need to be able to generate the sequence. And so I should have blocked these out, actually. Let's make sure that we can generate the sequence for our mRNA. All right, so again, this is all about complementary base pairing. So C complements G, then C, then G, A, G, U, right? Because for mRNA, it should be U. G, G, A, and then U, A, C. All right, so given a template strand, you should be able to tell me what the mRNA sequence would be that's generated from that template strand. Okay, so step one that we want to be able to do. Step two, what amino acid what tRNA anticodons are going to recognize this gene right here, this mRNA strand? So again, now you're just being complementary to this particular strand, but again, it's RNA, so you need to remember to use U instead of T. So this would be this following complementary strand. Again, you have G complementary to U, or I'm sorry, G complementary to C, C complementary to G, A complementary to U, and U complementary to A. And then importantly, being able to use that table to generate the polypeptide sequence that corresponds to this gene. And if we look here, this table is the table of codons. So these are the mRNA fragments. These aren't the anticodons on the tRNA, these are the codons. So I would just go and match for each 3-letter combination. I need to find the corresponding amino acid, GCG. So let's look up our table here. How this is organized. First letter is in this column. Second letter is in this column. Okay, so again, what was it? GCG. So G, C. So that takes me to this block here. And I need GCG. Actually, all of them correspond to alanine. So the first amino acid in this sequence would be alanine. Next is AGU. So go here, get rid of my markings. So A, G, that takes me to this column right here, right? A, G. AGU would be a serine. GGA, here's G, here's G. So GGA is a glycine. And then UAC. Go over here, UA, so this is UAC, would be a tyrosine. All right, so that's how I would use that information on my constants page to figure out what the polypeptide sequence is going to be given an mRNA sequence. All right, same thing. They even make it easier for you here. Let's do one more that's slightly more difficult. This would be slightly more tricky if I just gave you an mRNA strand like that. So here the question was, write the amino acids that are coded by this section of mRNA. All right, so this is just saying, here are these mRNA codons, what amino acids are coded. But what if I said, here's this mRNA fragment, what would be the polypeptide sequence that would be generated, right? So here's what's floating around in the cell. A ribosome grabs onto it. What is the actual polypeptide that you're going to get? For this, you would need to first find, you wouldn't just start reading right off the bat. You have to first find the start codon. And notice that the start codon always corresponds to a methionine. So methionine is the first amino acid that's added. So I need to find that start codon before I know what the actual polypeptide sequence is going to be. I need to go move along until I find an AUG. So here's my problem. GAC, that's not an AUG. ACU. CUA. Notice I'm moving one fragment at like one 3-letter fragment at a time. Okay, so I'm yeah, one 3-letter fragment. I'm moving one how do I say that? One nucleotide over and then looking to see if I have that start codon. Because it's only after I have found that start codon that I'm going to be reading in 3 nucleotide increments. Until I've found that, I don't know what's called the reading frame is. So I'm going to keep on moving along until I have found, here's my AUG. Here's my start codon here. This tells me my what's called my reading frame. So again, AUG was my start codon. I found my AUG. This tells me, again, what's my reading frame, which is now I'm going to start reading this nucleotide sequence in these 3-letter chunks. All right, so let's just get it started here. So first one, again, AUG corresponds to a methionine. That's the first one. So now I have to find my second codon is AAC. So using my chart A, so AAC is an asparagine, ASN. UGU, going to back to my constants page, UG, so UGU is a cysteine. GUA, GU, so GUA is a valine. Okay, and how do I know when to stop here? I have to keep going just like the ribosome would. I have to keep going until I hit a stop codon. So I'm just going to keep on going, keep on trucking along here. Here's my next one would be this AGA. I look it up. AGA is an arginine. And then I get to UAA. UAA is actually going to correspond to a stop codon. So this does not have an amino acid associated with it. This is the tRNA that binds and breaks the whole thing apart. So at that point, I have reached, this would be my stop codon, and so I'm done. This is the amino acid fragment that would be generated from this mRNA strand. Notice that these nucleotides at the beginning didn't matter because I hadn't hit my start sequence yet. Once I hit my start sequence, my start codon, that showed me my reading frame, which 3 codons I should be reading. And then once I hit the stop sequence, I'm done. Again, these nucleotides at the end here didn't do anything for me, right? Because they were after the stop sequence.